So, horizontal gene transfer. What is that? Organisms need to adapt to the environment, right? And adaptability depends on the ability of organisms to generate significant genetic diversity, okay? Higher organisms like plants and animals, they generate genetic diversity via sexual reproduction. Make sense? Bacteria don't have sexes. They don't have the concept of sexual reproduction. Mutations are not enough because they aren't fast enough, so there should be some other way to mix genetic information between organisms, and that other way is called horizontal gene transfer. So there are three types of horizontal gene transfer that we're going to talk about. The first one is transformation. Very widely used in biotechnological applications. It is a very simple idea. So you have live vegetative cells, it's a bacterial cell. And outside of the bacterial cell you have DNA, usually in the form of a plasmid. Plasmid is a circular DNA which is not essential for microorganisms. So it's not a genomic DNA. Does that make sense? It's not genomic. It can exist. If it's present in the cell, it may give the cell additional capabilities. Best illustration of this is the computer game that was popular quite a few years ago called BioShock, where main character could walk around and collect plasmids. And that gave that character some extra abilities. Okay. So bacterial cell just picks up DNA plasmid from the environment. That's it. The whole thing. Transformation. So why are we concerned about this? If you have a gene that conveys resistance to antibiotics, and this gene is on a plasmid, when cells die, the content is released into the environment. Read, plasmids are released in the environment. Other cells can pick up those plasmids. Does that make sense? And this will result in the spread of antibiotic resistance in the bacterial population. Are we clear? Second mechanism is conjugation, also known as the bacterial sex. So you have two cells, and one of these cells have a special plasmid, which is called F plasmid, or fertility plasmid. So the cell that does have fertility plasmid is called F plus cell. Any cell that does not can be called F minus cell. So what happens next? Genes on the fertility plasmid encode proteins that are necessary for the formation of pilus. Also known as sex pilus. Okay. Now, what happens next? The plasmid, look, the plasmid gets copied, and the whole thing, I'm going to put a dashed line to illustrate the whole thing. The copy of the fertility plasmid gets transferred from F plus to F minus cell. What do you have in the end? You have two F plus cells. It's like, zombie movie. Zombie, healthy person, zombie bites a healthy person, you have two zombies in the end. Make sense? Same story. Antibiotic resistance genes on the fertility plasmids are a huge concern because they will spread really quickly through the population.
What did you call the bridge between them?
Pilus, sorry, yeah, I'm going to put it here. It's a pilus. sometimes called sex piles, because it ensures the transfer of genetic information. We good so far? Now, the third mechanism is called transduction, and I left it for the last because it's kind of the longer one. Very interesting from research standpoint, from clinical standpoint, and contributes a little bit less to the transfer of antibiotic resistance. It still does, though. So this mechanism involves viruses of bacteria, bacteriophages. So I'm not going to go into a lot of details about the infection. We're going to get to the point right away, okay? Everything that's in blue belongs to bacteria. Everything that's in pink belongs to virus, okay? So you can see the codes. So let's take a look at the first type of transduction. You have a bacterial cell here, okay? And you have a virus. I'm going to make it very, very simple. So this is your virus particle, and there is viral DNA inside. Okay. Now, obviously, cell already has its DNA, okay, in the form of nucleoid. Okay, circular chromosome. We good so far? So what happens? Virus introduces its DNA into the bacterial cell, and the genes that are on this DNA are getting expressed. Okay? So they get expressed, more virus particles are produced. And some of these virus particles properly package viral DNA. Right? While all of this happens, cell becomes a mess. Cell basically is dying. Everything is being destroyed, including cellular chromosome, which gets chopped up. Okay? In this really, really crowded space where you may have up to 200 viral particles per bacterial cell, so it's really, really crowded, and accidentally some of those viruses, some of those bacteriophages will package bacterial DNA instead of viral. Does that make sense? What I usually compare it to is the end of a soccer practice in U9. Okay? When all these kids are running to their cars, they grab the backpacks, and after every practice, if you're a parent coach, you're going to get emails that my Johnny grabbed somebody else's cleats or somebody else's backpack or somebody else's, because they don't really pay attention. Does that make sense? So this is exactly the same. This virus as well, they're not leaving. I shouldn't make them anthropomorphic, but I will do it anyway, so I like it. And they just grab accidentally the fragment of bacterial DNA. Does that make sense? Then eventually, these virus particles, they depart. Okay, so this particle will kind of go on its way, and it will inject now bacterial DNA into some other bacterial cell, and there you go. You have a gene transport from one cell to another. Does that make sense? Good? Make sense? Okay. Now, this type of transduction is called generalized. And you can see that I was talking about what happens to the cell in the context, and the cell will die, the cell will die, the cell will break down. So the viruses, the bacteriophages that kill the cell almost immediately, we call them lytic bacteriophage. Any questions about the generalized transduction? Good? Now, there's another option, which is called specialized I'm going to give it a name in a second, but first I want to discuss the mechanism. So what happens in a specialized transduction? You have bacteriophage here, okay? And just like the previous time, DNA gets injected into a bacterial cell. But look what happens to viral DNA this time. This time, virus doesn't kill the cell right away. Instead, the DNA of the virus gets inserted into the DNA, into the chromosome of a bacterium. Okay? See that? Now it's inside. Does it kill the cell? No, it doesn't. The cell is actually absolutely fine. Are we clear so far? So cell will live with that what's called prophage. It will live and live and live and replicate and is going to be fine until the cell experiences some kind of a stress. Under that stress, this viral fragment, that prophage, will be excised. So basically molecular scissors and nucleases will chop it out and remove it. Does that make sense? Here's a little bit of a problem. Those molecular scissors are not particularly precise. So what you end up with is a viral DNA which on its tail carries a little fragment of bacterial DNA. Does that make sense? Just accidentally. This is a completely random stochastic process. So once this happens, the viral genes are going to be expressed. You're going to get new virus particles. Okay? And one of those virus particles will package that bizarre DNA that is a chimeric between the virus and the bacteria. All right? So now you have a next round of infection. Are you with me so far? The next round of infection. So in this next round of infection, this bacterial fragment will be transferred to another bacterial cell. Okay, does that make sense? So in this case, it grabs the wrong luggage. In this case, virus grabs the right luggage, but with its own luggage, it also collects some stuff from bacterial chromosome. Good? So this is called specialized transduction. And the bacteriophages that do it are called lysogenic. This name reflects that they do not cause the lysis right away, but have a potential to cause it. Okay? Any questions before we move on to other stuff? We're good?